Welcome to Planetterrian Daily, episode 107, coming to you on July 1st, 2026. Here's what's new in science, health, and longevity research. Blood circular RNA panel outperforms pTau217 and amyloid-PET for predicting Alzheimer's progression. Researchers tested a model built on 34 circular RNAs measured in blood across large cohorts. The panel predicted progression to symptomatic Alzheimer's disease and outperformed both pTau217 assays and amyloid-PET imaging. The work adds a blood-based option for early identification of individuals at risk. Because the signature is detectable before clinical symptoms, it could support earlier enrollment in prevention studies. Next steps include validation in independent longitudinal cohorts. A few episodes back, the show followed new Alzheimer's biomarker efforts in neuroscience research. Today's panel moves that forward by delivering a measurable signature that separates high-risk individuals earlier than current imaging or tau measures. Now, shifting to a very different area of research. An individual participant data meta-analysis pooled 51 randomized trials to examine cardiovascular outcomes under prolonged blood pressure lowering. The results challenge the prior assumption that extended treatment duration produces progressively larger risk reductions. The analysis focused on major cardiovascular disease events rather than surrogate markers. No additional benefit accrued beyond the initial treatment period captured in the trials. Future work will examine whether specific patient subgroups show different duration patterns. Both stories refine how we time interventions. Next, we turn to how pre-existing immune tone shapes what happens once an infection actually begins. A controlled human influenza challenge study profiled local and systemic immune responses in participants. Individuals with heightened innate cell responsiveness before infection were more likely to develop symptoms once exposed. The finding separates pre-existing immune tone from the adaptive response that follows infection. It suggests that baseline innate reactivity may shape clinical outcome more than previously modeled. Follow-up studies will test whether modulating this tone alters infection severity. From immune tone in infection, we move to mapping how gene networks control cell state in healthy stem cells. A genome-scale CRISPR interference screen generated a regulatory map across human-induced pluripotent stem cells. The atlas identifies functional relationships between transcription factors and target genes at pluripotent state. It supplies a reference dataset for dissecting how gene networks maintain or exit pluripotency. The screen covered thousands of genes and produced quantitative perturbation effects. Researchers can now use the map to prioritize candidates for directed differentiation protocols. Gene network maps like this one highlight how cells manage specialized machinery. A new study shows one parasite species solved the same problem by splitting a core mitochondrial function across 3 distinct proteins. Most people picture mitochondria as having one standard protein insertion machine that handles every membrane complex the same way. In reality, some organisms run multiple specialized versions of that machine, each tuned to different complexes. Right now, inside your own cells, the OXA1 insertase is threading newly made proteins into the inner mitochondrial membrane to keep oxidative phosphorylasin running. Trypanosomes carry 3 distinct OXA1 paralogs. Losing just one selectively dismantles either complex I and 4 or complex 3 and ATP synthase. One memorable detail is that a single parasite species evolved this 3-way split to manage a membrane proteome far more divergent than the one in human mitochondria. The practical takeaway is that future therapies aimed at mitochondrial disorders may need to target specific insertase paralogs rather than assuming a single pathway can be modulated uniformly. Specialized molecular machines also appear outside mitochondria. A new nanobody turns a common fluorescent tag into a multifunctional handle for imaging and manipulation. The single-domain antibody SDAB-MNG binds M-neon green with 0.39 nanomolar affinity, revealed by a 1.26 angstrom crystal structure. One reagent supports immunosuppressation, direct immunofluorescence, super-resolution imaging, and intracellular relocalization when expressed as an intrabody. In primary neurons, it visualizes synaptic vesicle transport. In zebrafish, it triggers cell autonomous depletion of an MNG tag transcription factor. The nanobody therefore converts a passive fluorescent tag into a programmable affinity module. Two theoretical physicists worked with the Claude system to settle a mathematical relation describing complex systems that had remained open for 10 years. The jamming conjecture had not yielded to earlier analytic methods. Their proof was published in the Journal of Statistical Mechanics, Theory and Experiment. The collaboration shows how large language models can assist formal derivations when directed by specialists. Additional uses in statistical mechanics are now under consideration. A conformal prediction approach named ConfDoc supplies atom-level uncertainty ranges for predicted ligand poses. It pairs graph neural network quintile outputs with split conformal calibration on 238 protein ligand complexes. Mean interval width fell by 57.2% relative to ordinary split conformal methods, while coverage guarantees stayed intact. The framework operates with any of 4 separate docking algorithms. It therefore offers a direct means to identify poses whose local reliability differs according to protein ligand context. A bacterial transpeptidase was adapted to attach biotin selectively to N-terminal diglycine remnants after protein digestion. The method recovered previously ubiquitated proteins and revealed an AU-twictination site on the E2 enzyme UBC13. Release can be achieved with guanidinium or by reversing the sortase reaction. A bifunctional peptide variant permits trypsin-compatible elution for mass spectrometry. The technique reaches non-canonical ubiquitination sites that antibody approaches often overlook. An international group isolated a new family of small proteins from Robinia pseudoacacia that instruct its bacterial partners to fix nitrogen while leaving the symbionts intact. The mechanism, reported in Science Advances, differs from the strategies used by annual legumes. The peptides maintain bacterial viability and redirect metabolism toward fixation. The observation broadens knowledge of how woody perennials maintain long-term symbioses. Researchers plan to search for comparable peptides in additional tree species. Molecular biologist Simon Bolton's laboratory maps how cells sense and mend DNA that becomes exposed during replication. Loss or mutation of protector genes leaves segments of the genome open to damage. The resulting map identifies repair pathways that become indispensable once protector activity is removed. These synthetic lethal connections are being examined as possible points for intervention. Ongoing screens continue to locate further context-specific weaknesses. A Lawrence Berkeley National Laboratory researcher ran a simplified hadronization model on a remote IBM quantum processor containing 104 qubits. The simulation reproduced string breaking, an essential stage in forming hadrons from quarks. The findings were published in Physical Review D. The result indicates that current quantum hardware can already address selected particle physics processes outside classical reach. Larger simulations remain the next stated target. Molybdenum disulfide was shown to generate methyl peroxide and additional liquid oxygenates from methane at temperatures below 100 degrees Celsius. The study, appearing in Advanced Functional Materials, required only modest alteration of an industrial catalyst. Methyl peroxide functions as a precursor to methanol, a dense liquid fuel. The route avoids the high-temperature processes that currently dominate methane upgrading. Scale-up and selectivity remain the next reported objectives. Grassland plots were exposed to the combined effects of warming, elevated CO2, and drought rather than to each factor alone. Published in Science Advances, the work showed that drought-driven losses in carbon uptake grew larger under the simultaneous future climate conditions. Earlier single-factor trials had therefore underestimated the strength of the interaction. The Innsbruck team measured the extra reduction across the full set of drivers. Extended field trials will test whether shifts in plant community composition change the size of the effect. These molecular tools help us measure and control cellular processes with greater precision. New findings keep refining how we measure risk and mechanism at cellular and system scales. Next time, we'll be watching for how the submillimeter array's new rapid response system performs on additional gamma ray burst events. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. OmniView, a balanced daily world news briefing that takes every side seriously. Find every show free at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.